Hello, and welcome to this Microbiology Bio 203 video lecture from Chapter 1 of your OpenSTAX book, An Invisible World. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. Learning outcomes for this chapter include being able to define microbiology, list the milestones in the history of microbiology, and explain the basis of classification of microbes. We begin today by exploring just what microbiology actually is all about. Microbiology is the science that studies microorganisms. What is a microorganism, you might ask? Simply put, it's an organism too small to be seen with the unaided eye. A variety of subdisciplines has grown out of our study of microbiology. Subdisciplines by application include medical, industrial, and agricultural uses of microbiology. Subdisciplines based on the study of a specific microorganism include bacteriology and virology. Microbiology, in some form or another, has quite literally been around for hundreds, if not thousands of years. Fermentation was known and being used long before people really understood what caused it. Humans have been using high osmotic environments to preserve food for generations. High osmotic environments include salting and pickling foods, even smoking foods, to prevent them from spoiling. Disease has always been on the forefront of concern for human populations. Ancient hygiene practices and infrastructure of the Greeks and Romans, early concepts of immunity, all indicate a concern over the transmission of disease from a very, very early time. A man by the name of Marcus Tyrrenius stated, "Things we cannot see can cause disease." The first observations under a microscope of microorganisms was done in 1675 by Anton von Leeuwenhoek. Anton von Leeuwenhoek is not the first person to examine something under a microscope, but he is the first person to describe a living thing, which he referred to as animalcules. These animalcules were probably protists and bacteria that he was able to see with a very simple handheld light microscope. Edward Jenner is the first person in history to explore the concept of immunity. In Edward Jenner's time, smallpox was a big problem. Interestingly enough, he noted that in a village near his, dairymaids who had been exposed to cowpox appeared to be protected from smallpox. He hypothesized that a cowpox infection might then provide protection against smallpox. So, in an experiment, he inoculated a boy with cowpox fluid, then later challenged that inoculation with smallpox. Fortunately for Edward Jenner, since the child was his own, the child did not get smallpox and survived unscathed. The golden era of microbiology really begins with a man we might all remember or recognize, Louis Pasteur. Louis Pasteur was a French microbiologist who was alive from 1822 to 1895. Pasteur demonstrated that microbes are responsible for fermentation. Pasteurization, a process developed by Louis Pasteur, is one way of controlling the number of pathogenic microorganisms in any type of solution, and it involves the application of high heat for a short time. Pasteur also developed several vaccines. Robert Koch is also part of the golden era of microbiology. Robert Koch is a German microbiologist who was alive from 1843 to 1910. In 1876, Robert Koch proved that a bacterium causes anthrax. Robert Koch is responsible for the germ theory of disease and provided the experimental steps we now refer to as Koch's postulates to prove that a specific microbe causes a specific disease. Breakthroughs in microbial culture, although agar was Fanny Hess's idea and not recognized are also attributed to Robert Koch. Taxonomy. The science of naming and classifying organisms has been around, like the study of microbiology, for hundreds of years. The most notable name in this area is a man by the name of Carolus Linnaeus. His true name is Carl von Linné. However, he changed his name to Carolus Linnaeus because he loved Latin and he loved the Latinized naming system that he developed. The Latinized naming system that Carolus Linnaeus developed is referred to as binomial nomenclature. It is one in which each organism has two names, the genus and specific epithet. Organisms are given names now that are descriptive. They describe a color, a shape, or a location that they can be found. In some cases, organisms are given names based on the person who discovered them. Chemotherapy. The idea of chemotherapy is treating diseases with chemicals. This was something that was developed by Paul Ehrlich. Paul Ehrlich came up with the idea of a magic bullet, one which provided selective toxicity, which means that these chemicals would only be harmful for the pathogen and not to the host. Ehrlich's original work involved drugs for the treatment of syphilis, and later synthetic drugs such as sulfa drugs in the 1930s. Antibiotics. Antibiotics are produced by microorganisms to inhibit or kill other microbes. The first antibiotic, penicillin, was discovered by Fleming in 1928, pretty much by accident. A petri dish with Staphylococcus was left open, and the plate got contaminated with penicillium mold. Eventually, a clear zone shone around the mold, which led to the discovery of penicillin. Penicillin saved lives during World War II. It was mass-produced just in time. Alexander Fleming was hailed for this discovery. Less known are the contributions of his colleague and spouse, Mrs. Fleming. Modern biotechnology and genetic engineering. Biotechnology is the use of microbes to produce food and chemicals. This is actually a centuries-old science. Genetic engineering is the utilization of bacteria and fungi to produce a variety of proteins, including vaccines and enzymes. Missing or defective genes in human cells can be replaced through the utilization of gene therapy. Genetically modified bacteria are used to protect crops from insects and from freezing. CRISPR is a gene editing process which is based on normal bacterial processes and can allow us to make precise changes in DNA. Microbes and disease. The human microbiome is made up of microbes that are naturally inhabiting locations in the human body and ultimately contribute to our health. Pathogens cause diseases. Our normal microbiota do not. The diseases that are caused by pathogens are studied through epidemiology. Epidemiology studies how diseases are transmitted. Pathogenic factors contribute to disease. When there is an outbreak, we see an increased incidence of illness associated with a pathogen. Outbreaks might include foodborne diseases or emerging diseases such as COVID-19. Outbreaks can also include recurring epidemics like the flu. Environmental microbiology. Microbial ecology is a growing area of science in microbiology. In microbial ecology, bacteria are utilized to recycle such things as carbon, nutrients, sulfur, and phosphorus. Bioremediation is another growing area of microbial use, where bacteria are used to degrade pollutants. Phylogeny and the tree of life. Carolus Linnaeus is responsible for naming organisms using binomial nomenclature. He is the first person to establish a classification scheme for organisms based on their visible characteristics. An organism's phylogeny goes beyond just naming an organism and includes their evolutionary relationships. Currently, genetic, biochemical, and embryological considerations are used when determining whom is related to whom. In the 1970s, Carl Weiss used ribosomal RNA sequences to establish the three-domain system: bacteria, archaea, and eukarya. Here we see the three-domain system as based on those rRNA sequences. Again, it is made up of the bacteria and the archaea, and here we see the eukarya subdivided into the protists, which is sometimes referred to as Protists, the fungi, plants, and animals. Here we see an expansion of Carolus Linnaeus's work on binomial nomenclature, again emphasizing the idea that each organism receives two names, a genus and a specific epithet. Here are some examples of different organisms with their scientific names as given by Carolus Linnaeus. Remember, these scientific names are meant to be descriptive. They describe an organism's appearance, its color, shape, or maybe location where it can be found, and in some cases, the names that they have honor the individual who discovered them. The bacteria. The bacteria are all prokaryotic organisms. As a prokaryote, they have peptidoglycan cell walls, divide by binary fission, no mitosis, and they lack a true nucleus and any form of membrane-bound organelle. They do have a variety of energy sources. Some use organic chemicals, and others use inorganic chemicals, even the sun. The archaea are also prokaryotic. They, however, have a cell wall that is different than the bacteria. They lack peptidoglycan. They tend to live in extreme environments and include the methanogens, extreme halophiles, and extreme thermophiles. Most archaea can only be found in incredibly harsh environments. The Burgess manual is used to classify different bacteria and archaea. The Burgess manual is based on genetic, biochemical, and immunological characteristics of these organisms. Gram staining is a differential stain that's based on cell wall properties that helps us differentiate one organism from the next and ID it using the Burgess manual key. Protists. The protists include any and all organism that quite simply we don't know what else to do with. In this case, we'll look at the alga. The alga are sometimes referred to as the plant-like protists. Informal grouping of eukaryotes that are not plants, not animals, and not fungi make up the alga. Alga have cellulose cell walls. They use photosynthesis for energy. They produce molecular oxygen and organic compounds and are not pathogenic. Probably the most familiar alga to you, if you frequent the beach, is what most people refer to as seaweed. The protozoa are also protists. The protozoa are sometimes referred to as animal-like protists. These are the most complex one-celled organisms that exist on the planet. They are not considered animals because they are single-celled. They can absorb or ingest organic materials in a manner similar to an animal, but because of their unicellularity, they are excluded from the kingdom Animalia. They may be motile via pseudopods, cilia, or flagella, and many are human pathogens. Fungi. The fungi have chitinous cell walls. They use organic chemicals for energy. Molds and mushrooms are the multicellular varieties of fungi, while yeasts are the unicellular varieties. Molds can also produce antibiotics. Helminths. Helminths are multicellular animals. Helminths are parasitic flat and roundworms. They often have microscopic stages in their lives. Viruses. Viruses are acellular. They are described in this chapter on microorganisms as something that we will learn about in this course, but they are not a living thing. They consist of DNA or an RNA core. The core is surrounded by a protein coat. The coat may be enclosed in a lipid envelope. Viruses are replicated only when they are in a living host cell, outside of which they may remain dormant for generations. Prions. Prions are sometimes thought of as infectious particles. They have no nucleic acid, only proteins. They were first described by Prusiner, who won the Nobel Prize for them in 1998. The prion protein is an altered protein which makes a healthy molecule to change and become a prion. Prions can accumulate, form fibrils, and cause disease. A great example of a prion-caused disease is mad cow disease. And this concludes our coverage of Chapter 1 from your OpenSTAX book, An Invisible World.